to our distinguished speaker, all the respected guests, dignitaries and delegates on this virtual platform. On behalf of LM College of Pharmacy, I, Divya Teli, Assistant Professor, Department of Pharmaceutical Chemistry, welcome you all for the 5th webinar of the webinar series, Science of Science in the Spotlight. The speaker for today's session is Professor M.I. Yadav, sir, Director R&D, Parul University, Vadodara, Founder, Dean, ex-Head and ex-Professor, Faculty of Pharmacy, the MS University of Baroda. Today, he will be delivering an expert talk on drug discovery, whose prerogative and beyond. We are highly thankful to sir for accepting our invitation. Now, I would like to invite our principal sir for the welcome address. Let me briefly introduce him first. Dr. Mahesh T. Chabare sir belongs to pharmaceutical chemistry background. Talking about his academic achievements, he has around 68 research publications, 11 review articles, and 10 patents to his credit. He has a research grant of rupees 83 lakhs, CSR fund received from industry worth rupees 9.75 crore, and grant of rupees of Fatal Incubation Center at our college. He has supervised around 135 MPharm students and 14 PhD students. Sir is nominated as Associate Dean for Zone 1 GTU for 3 years since 2018. He is also nominated as Senate member for NIPER Ahmedabad, member of Pharmacy Board of AICTE for 3 years, 2020 to 2023, member of Syllabus Revision Committee for PharmD course by PCI in August 2020. With this, I would like to request our principal sir to give welcome address to our honorable speaker and the participants. Please, sir.
Thank you, Divya. Respected Dr. M. Yadav, sir, all the faculty members, students, and participants. On behalf of LM College of Pharmacy, I heartily welcome you all to this webinar, which is a part of a webinar series which we have started under Science in Spotlight, where we invite the eminent speakers across the country for their expert talk and then to share their experiences in their field. So this is the 5th webinar where we were thinking about we should invite an expert who is expert in drug discovery. So without spending a minute, the one name came into our mind. It was only Dr. M. Yadav, sir. And I call him that, sir, we are organizing such webinar, and he immediately accepted the invitation to share his experience in this. He has a vast experience in drug discovery process. And the topic which he has chosen is very, very right, and it's a need of time. We talked a lot about drug discovery. Many scientists are doing so, but now we have to think of whose prerogative and beyond. And I'm sure the kind of expertise he has, he'll surely share his experience, his thoughts, and ignite some spark to the budding pharmacists or budding researchers to think in that line. LM College of Pharmacy is the premier pharmacy institute of the country, which was established in 1947 by a very renowned philanthropic trust, Ahmedabad Education Society. Being the premier pharmacy institute, it has played a very important role in establishing pharmacy profession, not only in Gujarat, but across the country. It has produced very renowned pharma entrepreneurs, officials, academicians, scientists, which are spread across the world also. We have established or we wish to inculcate always a culture of innovation and invention in the students or even the scholars. To doing so, we have all platforms set in our institute. We have the Innovation Council approved by the MHRD. We have the Student Startup Innovation Policies Nodal Center approved by the state government. And we have the Atal Incubation Center, which is given by the Atal Innovation Mission, Niti Aayog. Now, when we are talking about the innovation and invention, obviously we have to think of a quality research. And to, again, I would say, inculcate the culture of quality research, not only in the students, even in the supervisor can always think about in that line only. We have started this webinar series, and part of that, this is the 5th webinar. And Yadav sir is always known for a high quality research. And obviously in his talk, you will find that he's doing so. And surely, as I said earlier, he will spark or ignite some spark to the budding pharmacists and the scholars for doing good or a quality research in coming years. I welcome and all the participants to this webinar. Thank you.
Thank you very much, sir. We at LM College of Pharmacy under IQAC and Research Committee are organizing a webinar series of Science of Science in the Spotlight with an initiative on quality and ethics in research every month at our institute. Developing a new drug from original idea to the launch of a final finished product is a very complex process, which can take 12 to 15 years on an average and cost of excess of $1 billion. Across the globe, most of the pharmaceutical majors and academic institutions are continuously in the lookout for developing a new molecule, which could be used as medicines to address various ailments. The Indian government is also working on various schemes to support medium-scale pharmaceutical companies and academia to grow to the next level. One of recent examples for academia is Drug Discovery Hackathon 2020, an initiative took up last year by MHRD Innovation Cell for COVID-19 drug discovery. As a result, we can say that drug discovery is not a prerogative of big pharma companies nowadays. So today, Professor M.R. Yadav sir will take us through various facets of drug discovery, whose prerogative and beyond. Now, without delaying the session further, with few announcements, we'll start the session. The attendance link for this session will be shared in the chat box, and the participants are requested to enter their questions in the chat box, which will be addressed by our speaker at the end of the session. Proceeding with our session, we have Dr. Anuradha Gajjar ma'am with us as the chairperson for today's webinar. So here I'll take the opportunity to introduce ma'am. Dr. Anuradha Gajjar ma'am is a professor and head of the Department of Pharmaceutical Chemistry and Quality Assurance at LM College of Pharmacy. She has pursued her B.Pharm and M.Pharm from Principal K.M. Kundanani College of Pharmacy, Mumbai, and PhD from Institute of Pharmacy, Nirma University. She started her career in academics in 1996 with KB Institute of Pharmaceutical Education and Research, Gandhinagar, where she contributed until 2003. Then she has served at Institute of Pharmacy, Nirma University, and Ramanbhai Patel College of Pharmacy, Charusat University. At RPPC Charusat University, she was entrusted the responsibility of NEC accreditation as coordinator IQAC in addition to her teaching assistant. The university earned the distinction of being the first private university in the state to receive A grade from NEC in the first cycle within a short tenure of 7 years since inception. She had been officiating as principal Ramanbhai Patel College of Pharmacy since July 2016. In June 2018, she joined LM College of Pharmacy as professor in the Department of Pharmaceutical Chemistry. She was awarded Most Innovative MPharm Thesis Award at the national level guide category for the thesis title Design, Synthesis and Pharmacological Evaluation of 2-amino-thiophene derivatives as antibiotic agents in the field of pharmaceutical chemistry for year 2012 to 13, instituted by Troika Pharmaceuticals and Department of Science and Technology. She has guided 10 research scholars for their doctorate and more than 40 PG students for master degree. She has, to her credit, more than 40 publications and more than 80 presentations at various forums. She has delivered more than 30 technical and motivational talks at various events. She has rendered professional services to a number of renowned universities of the country, as well as government organizations like DST, AICTE, and NEC. I welcome Dr. Anuradha Gajjam ma'am to chair this session. Over to you, ma'am.
Thank you very much, Divya, for the kind words of introduction. And it is indeed an honor for me to chair the session being delivered by Yadav sir. I take this opportunity to introduce a renowned personality like Yadav sir to formally of Research for Development, Parul University, Baroda, Gujarat. Earlier, he has a long span of academic career, and actually he has held many posts of various organizations. Earlier, he was a UG at Faculty of Pharmacy, the MS University of Baroda, Head Department of Pharmacy and Dean, Faculty of Pharmacy, the University of Baroda, Vadodara. He is the founder of research experience spanning patent applications to his credit and 6 books and book chapters. Many of his work has been contributed or has been presented by his research team various research modeling and drug and synthesis of inhibitors and anti-hypertensive agents and site-specific chemical deliver more than 34 students have been awarded PhD students guided by him. He has handled for evaluation of the. He has been the advisory board of referee most
some internet problem.
Madam, hello. Madam, you are muted.
Now am I audible?
Yes, ma'am. In between, your voice was broken.
Okay. I'll just. Dr. Yadav has been awarded with Teacher of the Year 2010 award by Association of Pharmacy Teachers of India and Eminent Teacher of the Year 2013 by Association of Pharmacy Professionals. Other awards received by Professor Yadav include by University Institute of Punjab University 2015. Sir, example by successfully using GPAT in 2000 as the chief coordinator for the first time in India. At national level, Dr. Yadav his body is also on the of many universities. An illustrious sir, his talk on drug discovery, prerogative, and beyond. We are waiting.
Hello.
Yes, sir, we are able to hear you.
Okay, so can I start?
Yes, sir.
Okay, thank you, Dr. Anuradha Gajjar, for a very nice introduction. And thank you very much, Professor Chhabria and Ms. Divya Patel and the team for inviting me to be one of the resource person in your series of lectures. Before I start, let me share the screen first. So is the screen visible? Hello? Am I
Yes, sir.
audible?
Yes, sir.
Yes, sir. Yes,
Yes,
sir.
sir.
It's visible.
And
Yes.
what about the screen? Is it visible?
Yeah, it's visible.
Yes, sir.
Okay. So this is the title which I gave. Just a minute. This bar is interfering in the screen, so let me put it at some suitable place. Okay. So it is, I would say, a matter of pride that LMCP educational initiative is being taken by LMCP on quality and ethics in research. And a webinar series on science of science in the spotlight has been arranged in the form of a series of lectures. But there is an irony in both the titles I find. See, when you talk about quality and ethics in research, people in audience, they take it sort of very philosophical. And because it's a very simple type of a thing which can easily be understood, people understand it, but unfortunately they don't practice it. See, it's something like everybody knows honesty is the best policy, but then people think that it is not practicable. When it comes to science of science in spotlight, here my experience is one should talk hardcore science, right? And when you start talking hardcore science, people don't understand. And because they don't understand, they don't practice it. So either way, my experience has been, if you are too simplistic, people take it to be sort of a preaching. They take it to be so simplistic. It's not worth practicing. And when it is tough to understand, because they don't understand, they don't practice. So thinking on these lines, I thought what should be the title on which I should speak. So I thought let it be a mix of both of them. Let me share my personal experience and thoughts. So that means whatever I'm going to speak is partly science, may not be hardcore science, and partly sort of philosophy. And before I start, I mean, why did I give this particular title? There are two incidents which happened, which in fact made me to think about this particular topic. One incident, it happened at the beginning of my career as a researcher. And the second incident, which is recent discovery of COVID-19 vaccine, you can say it has happened at the fag end of my career. Let me first narrate this incident which happened at the beginning of my career. I was doing my PhD, and almost at the close of the experimental part, there were certain reactions which I was to carry out, but the facilities were not there in Punjab University, Chandigarh at that time for carrying out those reactions. So my supervisor, Professor Harkishan Singh, he was well acquainted with the director of CDRI, Professor Dr. Nityanand. He told me that you go to CDRI and complete your experimental work. So when I went to CDRI, I was assigned to work with a senior scientist in CDRI. So he called me and he asked me what are the things which you want to perform here. I narrated my complete scheme, why I'm doing and what type of reactions I'm supposed to carry out. After listening to that, he immediately posed a question. Why you are carrying out drug research? This is none of your job. This is our job. And it was a blow to me because I never thought on those lines that drug research is not the job of a pharmacy person, maybe from any field, maybe medicinal chemist, maybe pharmacologist. So, and later on, after that I'm talking somewhere around 84, I have met n number of people who went on claiming that drug research is only their field. And out of them, number of them are biologists, pharmacologists, even pharmacy people. Everybody claimed that it is their field and it is none of others' business to poke their nose into drug research. So I thought, let me clear some doubt in the minds of the young generation, whose prerogative is to go for drug research. Now, when we go for drug research, we now have to bifurcate into broadly two categories: small molecule drug discovery and macromolecules or biologicals. When you take up small molecule, we normally teach to our PG students. Drug discovery always starts with lead identification, later on followed by lead optimization and other steps. When you take lead identification, what are the different ways and means by which you can identify a lead? In fact, you know that there is no set procedure for identifying a lead. People have identified leads by different means, number of them, even accidentally also, which we call as serendipitous discovery. But a very important contribution is being given by the phytochemist. As you know, so many drugs have originated from natural origin. So phytochemists who are basically pure chemists, they investigate the phytoconstituents present in the plant. And these phytoconstituents then, can turn out to be lead molecule, which can be optimized to be very good drugs. And you'll find that hundreds of examples are there in the drug discovery where the leads have originated from phytoconstituents. Normally what they do, they isolate the product in pure form, submit it to the pharmacologist, pharmacologist would screen them for biological activity, and ultimately some new molecule will be coming out as a drug. So there is a very important role of pharmacologists also. Can you say that pharmacologists are also not involved in drug research? So they have the only prerogative? No, they have also a very big role, because without the screening of the drugs, you will not come to know what type of activity they have. Then synthetic chemists, we know that, in the past especially, how, lot of drugs have been discovered. Large number of drugs, they have synthetic origin. So what normally synthetic chemists used to do, because they are possessing expertise in the synthesis, they will go on synthesizing the molecules of their choice, submit them for the screening. And in this process also, of course, it's a very lengthy and very time-consuming process, but number of drugs have originated from this approach, which may involve simple synthesis and blind screening. Now, computational chemists have played a very, very important role. Nowadays, you have drug designing approaches which are totally, you can say, simulated techniques, which basically are based on the computational chemistry or quantum chemistry. And you can very well imagine that maybe around 15 to 20 years back, leave aside pharmacists or biologists, even pure chemists never thought of touching computational chemistry or quantum chemistry, because the calculations involved, whether it is molecular mechanics or whether it is semi-empirical methods or ab initio. Ab initio used to be one very, very tough, because very lengthy Schrodinger equation need to be solved. So in fact, nobody dared to enter into the field of computational chemistry. Now, with the advent of very fast computing machines and high-speed graphics, we have softwares which will perform the calculations in a fraction of seconds or minutes. And the results are very easy to interpret, because the results would be displayed in the form of graphics, which you can very easily understand. So with the involvement of computational chemists, now it has become so easy that even you'll be surprised that, leave aside pharmacists and chemists, even biologists have become very friendly to the computational chemistry aspects. You will be surprised that nowadays more of the drug designing is being performed by bioinformatics people. Why is it so? It is because of the advent of the progress made by the computational chemist. And all these are simulation techniques, which gives a lot of good idea about the direction in which we should proceed for the drug discovery. When it comes to identification of the target, see, we teach different approaches in drug discovery. And if you can identify the target, it becomes a bit easy to design and discover a drug. identification of target has become so important that I think you may be knowing that the human genome mapping was performed purely you having this particular aim in mind, that once we have the complete sequence of human genome, we'll be able to know the different functions of the genes and what type of proteins are being expressed, what type of role they have, what type of proteins which are expressed will have a role in the physiological action in the body. So with that particular aim, the human genome mapping was performed. Unfortunate part is that although the project got over maybe somewhere around 2001, no one have the complete sequence of the human genome, but we still do not know the role of 99% of the genes, what role they play in our body, although their sequences are known. But what I mean to say is, the biologists are playing a very, very important role in identifying various targets which we can use for designing of the drugs. See, you just think that if suppose the structure of the enzyme or receptor is not available in the PDB database, then there will not be any 3D structure. There will not be, there won't be any simulation. There won't be any docking experiment. All that is, it has become possible because biologists have, structural biologists, they have isolated the enzymes or the receptor. If the receptor is not being isolated as such, they have been expressed by biotechnologists in the animals in pure form, so that the 3D structures are known. And once the 3D structures are known, then they can be utilized for designing of the drugs. Similarly, biochemists have played very, very important role, right? So biochemists, we know that what type of reactions, the biological reactions on in our body, that is being decoded by biochemists. So a lot of things, and it's very, very important. You must be knowing that lot of drugs, they have originated by studying the biochemistry of our body. Like say, majority of the ANS drugs, right? Parasympathetic mimetics, sympathomimetics, their antagonists, then various hormones, anti-hormonal drugs, they all have originated because of the work of the biologists. And biotechnologists, I have already emphasized that biotechnologists, because they manipulate the gene, they can get the gene, they can isolate the gene, they can get it expressed, they can put it into the animal or into the bacteria, and then through expression, we can have particular protein or enzymes. They can be isolated, the 3D structures can be determined. So all that is possible because of the help provided by the biotechnology. All these specialists, they are playing very, very important role in drug discovery, right? And without their involvement, you cannot think of the pace which we have attained in discovery of drugs. It can be achieved. It is impossible. Similarly, once a particular drug is being identified, not drug, rather lead is identified, because lead might possess different side effects. So you would like to optimize it. And when we say that we want to optimize a particular lead, means basically we want to get rid of the side effects of the lead and increase the potency. Who are playing the role in lead optimization? Of course, synthetic chemists, because they have to chemically modify the lead molecule into different derivatives, so that not only side effects are removed or minimized, the potency is also increased. Pharmacologists are involved because without them, the screening will not be possible. And once a particular compound shows a favorable biological activity with potency and lesser of the side effect, toxicologists, they have to evaluate their long-term and short-term toxicity. Their involvement is very, very important. And once the pharmacokinetics and other things are being involved, the pharmaceutics, they have to formulate it. So obviously they are very important component or constituent of the drug discovery process. When it comes to biological therapeutics, that means basically drugs which are macromolecules or a combination of macromolecules. Like, you have vaccines, sera, biosimilars. Who is doing this job? All this is being done by either the microbiologist or the biologist. Without the involvement of microbiologists, vaccine, sera would be impossible, which a medicinal chemist can never do, right? Similarly, biosimilars, number of biosimilars are now in the market and the market is increasing for biosimilars. It is basically the work which is being performed by the biologist. And once a drug is being recognized, being identified, then it cannot be administered just like that in crude form. It has to be formulated in a suitable formulation, whether it is a conventional dosage form or whether it is a new drug delivery system. Here the involvement of pharmacy people is very, very important. But let me tell you, pharmacy people should not remain under the impression that they are the only people doing this job. You'll be surprised that especially in Western countries, this job is mainly performed either by pure chemists or by chemical engineers, right? So even this is not purely a prerogative of pharmacy people. All people are involved. So in nutshell, what I would like to say is that nobody can claim that this is the field in which nobody else should intrude, and it is their field only. Drug discovery is a process which is a multidisciplinary approach, and you require the help of people from different fields, right? Like say, you'll be surprised that there is, I don't know how many of you have read about the history of discovery of cimetidine. H1 blockers were known in the early 50s and 60s, but it was known that histamine has effect on the stomach also, which is not being antagonized by the H1 blockers, the antihistaminics. So a group of scientists started working on the basic molecule histamine, how to modify the histamine molecule in such a way so that the new molecule becomes an antagonist which specifically works on the receptor which are present in the stomach. There, the role of physical chemist, how the imidazole moiety present in histamine is being monitored and changed so that it will interact only with the S2 blockers, S2 receptor, not with the H1 receptor. So the role of physical chemist was very important in the discovery of cimetidine. So in nutshell, what I would like to say is that the drug discovery is a prerogative of everybody who can contribute in the process, right? So it should not be claimed that this is my prerogative and nobody else can, come into this field. Just a minute, this is not moving. Yeah. So, whose prerogative is it to do drug research? This can be equated with one sentence. An elephant will not be an elephant with just its tusk, trunk, and tail. It will be a half story. If you have just tusk, trunk, and tail, you cannot say this is an elephant. The drug will involve, the involvement of all these people, then only it can come out as a drug. And the second incident, which I was telling, recent discovery of COVID-19 vaccines. See, we have been studying and we have been teaching to the students all these years that a drug takes anywhere from 10 to 15 years from the bench coming to the market. And it involves huge amount of money. In fact, if I remember correctly, in 70s, it used to be said that a new drug molecule costs somewhere between 200 to 400 million. Then, when it came to 80s and 19, it was said that the involvement could be anywhere between 600 to 800 million dollars, right? And now it is being said that in this century, a new drug costs somewhere around 2 to 4 billion US dollars, right? And time, of course, it might have been shortened a bit, but 10 to 15 years is a normal range which is being taught to the students, and we also read this thing at different platforms. One day, maybe around 3-4 years back, I was in Chandigarh with my supervisor, Professor Kishan Singh, and we were talking on this particular aspect only about the drug discovery. And I asked him, sir, how much money was involved in the discovery of kendocuronium? Let me tell you, majority of you may not be knowing. Kendocuronium is a neuromuscular blocking agent which was discovered by Professor Kishan Singh group in Punjab University, Chandigarh in mid-70s. And earlier the name of kendocuronium was chandonium, because majority of the scientific reports, research papers, they are with the name of chandonium iodide. So this was the only drug which was discovered from an Indian university, and in fact it was approved also, but it was never marketed for different regions. So I asked, sir, how much money was involved in the discovery of kendocuronium or chandonium? Then he said, look, Dr. Yadav, the chandonium was discovered under a CSR scheme, and the cost of the scheme was 2 lakhs. That was in 70s. Of course, in 70s, rupees 2 lakh was a big amount, but it was not as big as it's being projected for the discovery of a new drug. And within this much amount, the drug was synthesized and evaluated. And the clinical trial, because it was a CSR compound discovered under the scheme of CSR, so the clinical trials were being performed at CDRI and King George Medical College, Lucknow. So you can add just a few more lakhs rupees more on clinical trials, maybe 10 lakh, 20 lakh. So if you take that figure, this new molecule may not have cost more than 50 lakh at the most, which is also highly projected, right? And we now say that a new drug cost somewhere 2 to 4 billion US dollars. And now talk about the duration of discovery. The duration of discovery still remains 10 to 15 years, but no less than 5 to 7 years. It is being written in number of places, no less than that. Now think of a new drug, you can say, for COVID-19 vaccine. This COVID-19 vaccine has been discovered within a span of 1 year only, right? How come is it possible? See, the difference between a small molecule drug and a biological like COVID-19 vaccine is only that for small molecule, you don't know what is the target which is going to be hit in the body. So you are totally blind as far as the target is concerned for which you want to make a drug, right? In case of COVID-19, the target is known, right? So you have to hit the retrovirus, the RNA or maybe the DNA which is being expressed in the body. So the target is known. So of course, less work you have to do as far as development of vaccines are concerned in comparison to the discovery of new small molecule. But this time period cannot be shortened to that extent. Now, two things comes very, very clear to my mind as far as these two aspects are concerned. Number one, the amount which is being projected for a new drug discovery is somewhere 2 to 4 billion US dollars is highly, highly exaggerated. And second thing, even the duration of time which is required for discovering a new drug, 10 to 15 years, is again highly prolonged projected span period. Because when you can discover a vaccine in a year, why can't a new small molecule be discovered maybe in 4 or 5 years? So I personally feel that these two particular aspects are highly blown by the Western people, Western researchers. So my personal feeling is that these two things are blown intentionally so that, you know, developing countries people, they don't enter into the field of drug discovery. And I have interacted with many Indian scientists also. They also feel that this much amount is not required for discovering a new drug. So there is something wrong with the projections which are being given in various journals and magazines as far as the drug discovery time period is concerned and as far as the money spent on discovering a new drug is concerned. So we should give a second thought for both these aspects. I personally feel that it is not as difficult as is being projected for discovering a new drug. It doesn't take that much time. It should not take. And at the same time, it should not take that much cost also. I think if the government systems, they help you, I think it is very much feasible to come out with a new drug in much less time period, in much less cost. So this is as far as this aspect is concerned. Now I come to the second part of my talk, and beyond. Beyond means basically, of course, I just wanted to give you a bird's eye view as far as the involvement of different people in drug discovery is concerned and the cost and the time period. Now, because as you know, I am a medicinal chemist, so I thought let me share some of the things with my new colleagues as far as the field of medicinal chemistry research is concerned. Because we are a group of teachers or scholars, right? How to excel in medicinal chemistry research? Because we have to perform a dual job. We are teachers in the first place, and then we also have to get involved in research in medicinal chemistry. So how to excel not only as a teacher, but also as a researcher? Now for this, in my view, we have to give advanced training in mainly in these fields which we have listed. See, at majority of the platform, wherever I interact with the teachers and students, I say that if you want to become a good, you should have three particular aspects very strong in your favor. What are the three aspects? One is the drug synthesis. That means you have to be abreast of advanced organic chemistry. See, you simply cannot say that basic organic chemistry is just enough for becoming a good medicinal chemist. Basic medicinal chemistry is, I would say, a necessity up to B.Pharm level. But once you go into the research field, knowledge of advanced organic chemistry is essential. And it has, so many dimensions. I personally have found that when I say we means basically we pharmacy people are badly lacking as far as the knowledge of advanced organic chemistry is concerned. Not only that, the conventional chemistry, basics are missing. There are a lot of advancement which has taken place. You may be knowing, you may be surprised that conventionally, see, a good chemist always thinks that CC bond formation is the real chemistry, right? Carbon to carbon bond formation, carbon to nitrogen, carbon to oxygen or heteroatom is very simple. Not much of the chemistry is required. That means even with basic chemistry you can do it. But the knowledge for making carbon to carbon bond is the real chemistry. And with conventional chemistry, number of carbon to carbon bond formation is not possible. It cannot take place. So there has to be an involvement of the chemistry of transition metal, the chemistry of boron, silicon, chemistry of phosphorus, sulfur. A lot of reagents are now available. And if we do not teach this chemistry to our students, see, they will be lacking in the advanced methods of organic synthesis, which are required for the, drug synthesis. So we have to keep abreast ourselves, and we have to make our evolving this chemistry of special elements like transition metal elements. Second thing, which is essential for a medicinal chemist, is the spectral interpretation. That means a good knowledge about the spectroscopy. Here again, I have found that majority of the pharmacy people, they lack this knowledge. And it is very unfortunate. This is what is happening even now. See, talk about spectroscopy. See, when you teach spectroscopy, there are different aspects. Like, what is the basic fundamental behind the technique? What is the instrumentation? And what are the different applications? See, we pharmacy people, or we as medicinal chemists, we are more concerned with the application aspects. Unfortunately, what we are doing, we teach a bit of the fundamental, and majority of the time we remain indulged in instrumentation, which is not our area. Even the instrumentation is not known. It is not very important. We don't miss anything much. What is basically required is good basic fundamental and then the application. Then when you ask any student about interpretation of any spectra, you give a student a spectrum of any compound and ask them, you give an interpretation, they fail. They cannot even identify a particular peak. Or if you give a compound and then ask them, tell me what are the peaks in proton NMR, what are the position and in what multiplicity it should appear, they simply fail. You ask them, what are the deuterium exchange studies? They fail to understand. And this is the basics. I'm not talking about the 2D or the higher studies of NMR, because now NMR spectroscopy has become so powerful that simply on the basis of NMR spectroscopy, if you have 2D spectra, different type of 2D spectra, you can immediately find out the structure. And let me again emphasize about the importance of spectroscopy, which I quote at number of platforms. I don't know how many of you know that morphine structure was tentatively given by Robert Robinson, right? Robert Robinson gave structure of morphine. And now look, morphine comparatively is a simple molecule. If you compare it with complex molecule, various steroids and others, it's very complex in comparison to morphine structure. But this morphine structure was given tentatively, not a full structure. He said it is a tentative structure, it may change. And it took almost 104 years for the scientists to give the structure of morphine, right? Since the time it was isolated and the structure was given by Robert Robinson, it took almost 104 years. What were the reasons why it took such a long time? And not only one person was involved, hundreds and thousands of chemists were involved in isolation in giving the structure, because at that time structure elucidation was performed purely on the basis of synthetic and degradative chemistry. They will degrade the compound, make derivative, again degrade them, make derivative, identify the degraded products, and then join them so that they can come out with this set. Simple reason was that those times only UV and at the most IR were available. The higher spectral techniques like NMR and the advanced NMR, they were not available. So it took 100 years to give a structure of morphine. Compare it with the structure of paclitaxel. Paclitaxel again, complex, much more complex than morphine, right? It took almost 11 years to give the structure of paclitaxel. It was much more complex than, and of course, spectral techniques like NMR were available, but the advances were not available, right? So it took much shorter time in comparison to morphine. But nowadays, the techniques are so powerful that you can come out with the structure of a compound, maybe if not weeks, maybe in days and weeks, right? So this is the power of spectroscopy, which unfortunately we are badly lacking. Third aspect is the drug design techniques. As I said, nobody dared to touch the quantum chemistry or you can say any of the techniques which were there in case of quantum mechanics or quantum chemistry, because so much of mathematics was involved, nobody could understand it. So very few people who were expertising in that particular field in quantum chemistry and quantum mechanics, they only were doing some job. Now with the advent of computers and graphics and all that, now it is sort of existing on our fingertips. Now everybody can do it. Drug design is one particular aspect which has been simplified like anything. You can perform even ab initio calculations, maybe atoms. Earlier, ab initio calculations could not be performed maybe for a few atoms, right? A molecule which is consisting of few atoms. So this is the power of the quantum chemistry, which has been now being utilized in the form of various simulated techniques like docking and all that, right? So this is also very, very essential as far as the medicinal chemistry research is concerned. And people think that drug design all alone will help you in discovering a new drug. No, that's not the case. Drug designing techniques will only assist you. They will only help you, in arriving at some particular structure. They simply give you direction in which direction to proceed, but they will never be successful purely on their own to give you a drug, right? So this is a very important technique which should be learned properly by the medicine chemist. Another aspect is the phytochemistry. See, as I said in the beginning of my lecture, that many drugs have originated from plants, from the phytoconstituents. And in fact, there was a time when drugs were originating only from plants, maybe in 60s, 70s, right? And then, what happened with the advent of an introduction of communal chemistry, this phytochemistry research declined. And it was observed that, see, this was minimum somewhere in beginning of this era, that is beginning of 2000. Majority of the people, they were involved mainly in communal chemistry, right? And this phytochemistry was getting neglected. And then it was found out that with this decline of phytochemistry, the number of drugs which are getting discovered is also declining. So it was felt that it is wrong to neglect phytochemistry. And here I would like to say one important thing for the pharmacy teachers and students. See, the very first AICT syllabus for B.Pharm was framed somewhere around 90, right? Maybe 91, 92, or somewhere like that, 92, 95. And because at that time I was in Punjab University, Chandigarh, majority of the teachers of the Punjab University, they were involved in the framing of AICT syllabus. Professor Murthy from our MS University, he was the chairman, and then number of people were there in the board for helping him to frame the syllabus. So we at Punjab University used to have a large number of meetings for framing of the syllabus in different branches, like in chemistry, cognosy, ecology, and all that, right? And I remember there used to be a fight among the teachers of chemistry and pharmacognosy. Pharmacognosy teachers should say that phytochemistry should not be taught by chemistry people because it is plant-based. So it should come under the purview of pharmacognosy and phytochemistry. We should teach it. Now, ultimately, what happened that I don't want to go into the details of this. Ultimate result was that phytochemistry was removed from the curriculum of pharmacognosy. And you see the result. It is functional chemistry, the phytochemistry is never taught. I don't know what is the status now in B.Pharm, but it was stopped being taught in B.Pharm. And fortunately, now I have heard that in M.Pharm it has been introduced. Otherwise, it was not existing. So such an important part of the drug discovery as subject is totally being neglected. And I don't know how many of you know that now in phytochemistry, the the identification of phytoconstituents or biologically active constituent has totally changed. It is not the old phytochemistry which is being practiced. See, in old phytochemistry, what we used to do, we will extract the plant, and then the extract used to be segregated by chromatographic techniques into individual components, purified, and then submit them for biological activity. So it used to take a very long time to identify a particular compound. And a number of times it happens that once you identify a compound, then it is known maybe at a later part that the compound is well reported. It's not a new compound, right? So all your effort which you have put into it, all goes waste. Now, this is not the approach nowadays being used. Now it is dereplication. See, earlier the approach used to be called as the silver bullet approach, right? Because you are aiming for a particular silver bullet. Now the approach is total different. We now want to adopt complete metabolomics. That means we want to find out all the constituents which are present in the minimum period of time. And for that, dereplication approaches are being used. All these approaches, now they are based on two particular aspects. One aspect which is very important is the advancement in spectroscopy. You will be surprised that now you have not only LCMS, you have LCNMR, you have LCNMR-MS, all three techniques joined together, right? So that means not only you get the mass of the individual component, you also get the complete spectrum, and not only one-dimensional, even you can get two-dimensional NMR, right? And you just imagine the miniaturization which has been done. You'll be surprised that a sample size of 5-6 microliter is enough for getting a good NMR, even getting good 2D spectra. And what could be the quantum of the material which should be there? Maybe around 10 to 20 microgram. See, when we were sending our NMR, the operator used was that it should be minimum 50 to 100 milligram of the sample which should be there if you want to get a good NMR. Otherwise, your NMR quality will not be good. Now you just imagine just 10 to 20 microgram quantity is enough to get a good NMR spectrum. So miniaturization has taken place to that extent, and so much change that for NMR you wanted only deuterated solvents. Otherwise, this spectra in normal solvent cannot be taken. Now NMR can be taken in water. Solvent has been totally made irrelevant. Now, how this has become possible? It has become possible with the introduction of statistics, introduction of advanced spectra which are available in our database. So all that has been integrated, and maybe it's a matter of days that you get the complete picture of the plant constituents which are present in a particular extent. So you need not even bother whether they are known or unknown. Just with the help of the computational techniques, you can find out what type of constituents are present, and if there any constituent is present which is unknown, its structure can also be decoded. Even the software have come which will give a structure of the unknown also without the involvement of the scientist. So advancement has taken place to that extent. So even without talking too much of the advancement, I would say that for a medicinal chemist, minimum these three aspects are very, very important. That means good knowledge about organic chemistry, good knowledge about spectroscopy, and the drug designing techniques. Next thing which comes, especially to a teacher, a new teacher who has joined, maybe who is a fresh or maybe who is interested in carrying out research, is generation of research idea. How to generate research idea? See, one basic thing is ideas do not crop up in vacuum, right? For that, there is no solution. If you think that you should not try to become a Newton, that when the apple is going to fall, I'll observe and then some idea will come to me. No, it's not going to happen like this. You have to read, think about whatever you have read, again read, again think, again read, again think. This is the only way by which you can generate some good idea as far as the research is concerned. Now, one can say that it's, as a teacher, am I audible?
Yes,
Hello?
sir.
Yes,
Okay.
sir. Yes.
Now, one can say that, we as teachers, we do not have time. How can we devote time for reading literature and all that, right? Because, and to some extent, I agree, because it's very unfortunate that majority of the teachers, they have been loaded with a lot of administrative duties. I don't know why, how this has come, but it is there. It's a fact. They have to teach to the students. They also have to perform some administrative duty. So they, sorry. So of course they will find less time in going through the literature. But there is no way out. You have to read some literature. Of course, I will tell you in the next slide, I'm giving some example. How can you utilize your teaching for your research? But again, there is no substitute as far as the reading of research articles in the literature is concerned. Reading of literature, review articles, books, annual reports, and attending conferences, right? Try to interact with the researchers to the maximum extent so that you can disseminate your knowledge. Some people, they think that, we should not take, or we should not participate too much in the conferences. We should not divulge our ideas. I don't believe in that philosophy. So some people are there afraid that, once they interact with the people, their ideas will be stolen and then, they will be copied and all that. Now, I don't think this is the case. Nobody has time for your ideas. Everybody is busy with their own ideas. So don't afraid. Nobody is going to steal your idea. you can even discuss what idea you have, because some people, they remain under the impression that once they discuss something, it will be copied by somebody else. The likeliness of that is very less, right? Of course, I don't deny that it can happen. It can do happen. But even if it happens, how does it matter, right? It is not very, very important aspect in my opinion. But interaction is very important because you always gain from interaction, right? So the important aspect for teacher is generate ideas from your classroom teaching. You may not have enough time to go through the literature. So how to generate idea? So you can generate your ideas from the classroom teaching. And I'm going to give some example for that. Extracting ideas from teaching experience and corroborating with literature. Here, of course, I will give the idea how it can be done. But again, you cannot neglect the literature, because let us say, even if you get some idea from your teaching, you still have to read the literature to find out whether this type of thing exists in literature or not. Sometimes it happens that whatever idea came to your mind, you will find that it is already there in the literature. So that, once it is there in the literature, then there are no takers for that. So keeping abreast of the literature is very, very important. Now, these example I am giving because, as I said, I am also a teacher, right? And just like you, I also never had, too much time to go through the literature, right? So, and at the same time, I would say that 75% of my research has been originating from my teaching experience. That means whatever I taught in the class, from that only I derived ideas, and all those ideas, they got translated in research and got published, right? Some example I'm giving. This is one example. Amino ester derivatives of acids, which basically is an extension of Prerogative. See, when I was in Chandigarh, that was in 80s, at that time, the Prodrug approach came into existence. And as majority of you know that what are Prodrug? They are the pro-moieties, or you attach a pro-moiety to a drug and it becomes a prodrug, which is biologically inactive. Now, of course, prodrug was not my area of research, because my core area of research in which I did my PhD was the steroidal chemistry. This prodrug was being practiced even in our department by some of the teachers. And what was the it was a sort of a hot cake. It was being taken up by number of other researchers because it was a very simplistic approach. The approach, especially for the NSAID, was that the NSAID will have a carboxylic group, and the partial toxicity of the NSAID is because of the local action of this carboxylic group in our stomach, which causes gastric ulceration. So simple approach was you block the carboxylic group with some grouping which will not be acid liberating, because carboxylic group will liberate the proton. So you convert it into a functional group which is not having a proton. That means you convert into some ester or an amide. And that was a very, very common approach which was being used for making products of NSAIDs. That means you convert the NSAID into an ester or an amide product, right? Yeah, I also knew about it because I used to interact with some of the teachers who are involved in this type of research work. I was teaching the ANS class of drugs, right? In that, the parasympatholitics or anticholinergic drugs. I think you also must have either read or must be teaching that this is a very important class of anticholinergic drugs. like even atropine belongs to this class of drug. Dicyclomine, cyclopentolate, then propenthyline, methenthrin, they all belong to this class of drug. Now, what are these drugs? These are basically amino ester derivatives, where you have the amino alcohol. This amino alcohol is interacting with an acid part, and this acid basically is derived from, you can say, acetylcholine, because in the rule of 5 in cholinergic drugs you study that if the rule of 5 is violated, the compound doesn't retain the cholinergic activity. And to violate the rule of 5, what you do, you increase the bulkiness of the acid part, that is acetic acid you remove with some other acid, right? So using that approach, large number of amino acid derivatives were prepared as anticholinergic drugs. So I thought, when we are making products with simple alcohols, why not to use amino alcohols to esterify them, right? Next slide will be more clear. This is because more of text. Next will be the chemical structures. So suppose this is the NH8, right, with the carboxyl group. You react it with the amino ethyl alcohol, right? Now this amino group may be uncyclized or may be cyclized, right? And then this is converted into an ester derivative, and theoretically it should become an anticholinergic drug, because this is what we study in case of the SCR of anticholinergics, that once an acid is reacted with this type of a grouping, it becomes anticholinergic. So now this is the case that you have an NH8, you have the amino ethyl alcohol, this may be cyclized or uncyclized, and this should be the moiety which should be formed. So I theoretically thought, that means this was simply an idea which came to my mind, that why to make simple esters as prodrugs, why not to make this type of esters which have an amino grouping. Now, this amino grouping will have a specific advantage over the conventional prodrugs, which will not have an amino grouping or a basic function, because the prodrug which were being prepared by NSAID, they were neutral, right? Like say if you attach it with methanol, ethanol, propanol, or all that, they will give you neutral esters. But here is a group of derivatives which are basic in nature, right? And as far as the NSAID is concerned, you can have any of the NSAID, like you can have indomethacin, you can have ketoprofen, you can have biphenyl acid, fluoroibuprofen, or naproxen, or 6-methoxyethyl acetic acid, any of them, any of these moieties. And in fact, we utilize all of them for making this type of the esters. Now, theoretically, once you make this, they will not be neutral, they'll be basic. And they will have advantages over the neutral compounds. What advantage they will have? See, once they go into the stomach, they will be, nitrogen will be protonated because it is a basic nitrogen. After protonation, it will be converted into a salt. Salt will not be absorbed from the stomach, right? So your physical contact of the drug with the stomach lining is totally removed. So there will not be any damage to the wall of the stomach. And once this salt passes into the intestine, the pH of intestine is basic, then it will be converted into the original base, and it will be absorbed because the molecule becomes lipophilic and will be absorbed. And once it is into the blood stream, then we have in our blood pseudocholinesterases. These pseudocholinesterases, they will break it, they will break the acid part, and then they will liberate the NSAID, and the NSAID is free to show its normal biological activity. So using this approach, we synthesize the compound, and then we evaluated them whether they do have anticholinergic activity or not, because possessing anticholinergic activity by this type of molecule is another advantage, because in the GIT, they will decrease the gastric motility as well as gastric secretions. So that is another advantage over the physical contact mechanism, right? So we evaluated all these compound for anticholinergic activity. We determined their pharmacokinetics, and all were found to be as per the hypothesis. Whatever we hypothesized, it was found to be correct. They possess anticholinergic activity. Of course, the quantum of activity was not as much as atropine, but they were still very potent. And the kinetics also showed that they were getting hydrolyzed by the serum cholinesterase very fast. So that was very, very positive, and a thought which came by teaching. And extending this particular thing, we utilize. Do I have time? Hello.
Yes, sir, about 5 to 10 minutes.
Is it limitation? Can I ask for more?
No problem, sir. Then we can continue.
Okay. Okay, I will shorten it. So this particular approach then we utilize for site-specific delivery also, right? So what we did, we made not only the esters with an acid of simple amino ethyl alcohol, but we took the tropine also, and tropine is converted into the esters of the type which I gave the structures earlier, right? And then along with getting a tertiary nitrogen of the compound, we also quaternize it. And there was a specific aim for quaternizing this nitrogen. Now, this type of compound, they become highly specific for binding with the anions. We came across a report that in cartilages, the number of or the anions, their cartilages are rich in anions. So we thought if this compound can be given parenterally, it is going to be localized in the cartilages, because having a quaternary charge will attract them towards the cartilages having high negative charge. So this type of compound we utilize for the site-specific delivery of the NSAID for the treatment of rheumatic arthritis. Here this ester group was basically NSAID, right? So once you give the compound parenterally, it is going to be localized, it is going to be transported to the cartilage where it is required, and then it will be deesterified. That means ester group will be removed and NSAID will be liberated, and NSAID then will show its biological activity. And this we proved through the radio binding ligand studies, that yes, this compound is, after parenteral administration, is getting localized into the cartilages, and there it is getting accumulated. So this simple approach, which we started with making anticholinergic drugs, incorporating NSAIDs with various amino alcohol esters and then tropine, we could make a number of papers having a site-specific delivery at the joints. And all these drugs, we converted into the tropine esters. This is another example I'm taking from my own experience, designing of dual receptor inhibitors. So I would like to cut it short because it's a long story. See, I was teaching antihypertensive drugs, and in the antihypertensive drugs, you have various categories of drugs like alpha-1 blockers, beta-1 blockers, and all that, right? And AC inhibitors, A2 antagonists. So I was teaching the alpha-1 blockers where you have prototype like prazosin, dexazosine, terazosin, and A2 antagonists like losartan and other compounds, right? So when I studied the SCR of these two categories of compound, I found out that in case of alpha-1 blocker, this 6,7-dimethoxyquinazolone moiety is an essential part. It cannot be removed. It has to be there if you want to retain alpha-1 blocking. Concern, the structural requirements are highly flexible, right? We could find out in the literature that this imidazole can be replaced by a number of other heterocycles. Even it can be simple acyclic grouping also. And this biphenyl tetrazole or biphenyl carboxylic acid can also be replaced by a number of other groupings. So we thought, why not to combine these two moieties so that you can have dual antagonist, which will act on the alpha-1 receptor as well as the A2 receptor. So we designed the compound retaining 6,7-dimethoxy-4-aminoquinazolone moiety and making lot of changes here as per the presence of various groupings present in losartan type of A2 antagonist. And this series, because theoretically we hypothesize that this type of molecule, they should prove to be dual inhibitors of alpha-1 as well as A2. And after making maybe more than 150 or more than that compound, we evaluated them for both the type of activity, and this was the compound which was found to be the best. See, the peculiarity of this compound is it has PA2 value against alpha-1 10.1. You'll be surprised that it is 100 times more potent than prazosin, because prazosin has 8 point something A2 value. And as far as the A2 antagonist activity is concerned, it is almost as potent as losartan, 8.83. Losartan has 8.2 or something, right? So this compound was dual acting, having more potency than alpha-1, that is prazosin, and equivalent potency to losartan. So this is another compound, and we had again number of publication out of this work, and very interesting. And you'll be surprised that one very important finding we divulged in this work was that not only these compound, even prazosin possesses dual activity. We as medicinal chemists, we never thought from pharmacological angle, but we were able to publish that work also, that we divulge in that prazosin not only is alpha-1 antagonist, it is also A2 antagonist. So if you are concentrating in your work, even you can have some very good finding in other field like pharmacology or related fields. This is another example I would like to share with you, because this I really miss, and it is a very big missed opportunity for me. Designing of vicinal diaryl heterocycles. See, in my PhD, I worked on heterosteroids. So this type of moiety we prepared, because at that time we were interested in making anti-progestational compound. Those days, in 80s, there was a lot of emphasis on contraceptives. So my project was basically to achieve or to get contraceptive compound which can act by antagonizing progesterone on the progesterone receptors. So we synthesize different compounds having steroidal moiety, but having an oxidizable moiety attached to some side of the steroid, right? And the moiety on which I worked was the oxidizole. Now, during that time, I came across this molecule, which is nothing but nimesulide. Nimesulide was introduced into American and European market in 70s, but then later on it was withdrawn and again reintroduced in 90s. So when it was getting reintroduced, this came into my knowledge that nimesulide is a very good antipyretic compound. So I thought, why not to replace this oxygen with a heterocyclic moiety? That means this oxygen has a particular function of having a particular electron density within these two rings. So that can be achieved by introduction of an oxidizable moiety at this position. That means you can try to make compounds like this or like this. You have two phenyl rings, like these two phenyl rings with different groupings. And in between, in place of oxygen, you introduce the oxidizable moiety. This was a thought which came into my mind just after my PhD. Why not to make this type of compound? But things were not very favorable, and I moved to Delhi from Punjab, Chandigarh, where there was no research environment at all. So I couldn't work on this particular idea. And then in 92 onwards, celecoxib, brafecoxib, all belonging to this category where you don't have oxidizable but some other moiety, but the system remains same. They started coming up. And when we worked at MS University on this project, we missed that opportunity of making this type of compound, because by the time we started making compound and we started reporting it, it also came into existence or came into known that the rofecoxib type of compound, they have serious side effects. So in fact, let me tell you that although we could publish maybe 2 papers related to this work, but majority of the work still remains unpublished. Maybe more than 2 to 3 papers, it still remain unpublished because the journals were not ready to take it because of the side effect being shown by rofecoxib. So this type of missed opportunity, I really regret. Had I started my work, it would have been a pioneering work in this particular field. Then in this one more aspect, I would like to say these are some example I have given from my own teaching experience, which got implemented into research. But let me tell you, if you think on these lines, these 2 aspects are very, very important. That is sorting out your ideas, assessing the feasibility and competence of publication. Assessing of the feasibility is very, very important, because whatever idea you have, you have to see that it is feasible to carry out the complete experimentation, maybe in your institution or maybe help of some of other institution. Let us say if you get some very good idea of making some anti-cancer compound and then you don't have facility, or maybe if suppose you get some idea of making antiviral compound maybe for COVID-19, but you don't have facility for evaluating the compound for antiviral activity, then what's the idea? It becomes a futile exercise. So assessing the feasibility of your idea is very, very important. Whatever idea comes to you, maybe through your research, you have to assess that it should be feasible, not only synthetically, but also the evaluation of the biological activity. Second very important aspect is the competence for publication. See, you also have to be sure that whatever idea you are going to translate into your research work should be publishable. Here I would like to give my own example. When I was doing my PhD work on steroids, that was in 80s, late 80s, you can say, because I started my PhD in 82 and then got awarded in 86. We found it very hard to publish the work, although it was a very good work, fantastic work, but nobody was ready to publish it. Reason was that the trend of research on steroid immediately declined after late 70s. In 60s and 70s, if you see all chemistry journals, they were full of research work on steroids. But then researchers lost interest in steroids, and then it became very difficult to publish the research work in good journals. So you also have to assess that whatever, because in research, it is a trend which matters. Like say, nowadays there is a big trend on working on anti-Alzheimer's, anti-Parkinsonism drugs, antiviral drugs. So you also have to see the trend. If the trend has declined, then even if you are having very good work, it will be difficult to publish it. So that's very important aspect I would like to emphasize and tell to my audience. I think it is high time. I wanted to share some of the ideas as far as ethics in research is concerned, but I think it may be too late. What do you say? Hello?
Yes, sir.
Hello?
Yes, sir. Yes, sir.
Should I stop?
No, sir, you can continue for a few more minutes, not an issue.
Okay, I'll just go through. See, quality and ethics in research is something which, again, I am not very positive. I'm very sorry to say that whatever work we carry out in our education institution, it has a very poor quality. And it is mainly due to inadequate training, which we need to improve. And another very serious aspect is indulgence in research misconduct. That means whatever research we are carrying out or whatever we are reporting is not fair. It includes lot of things, lot of fabrication or other things or plagiarism. And that, in my opinion, is because of low moral values or low moral ethics, which I'm very sorry to say that we have failed to inculcate high moral ethics and values in our young generation. Because of that, we are failing in these aspects. So I just wanted to share what is research misconduct and all that. It involves, your misconduct in posing or performing or reporting of research. What is the definition? Basically, it includes fabrication, falsification, and plagiarism. And plagiarism is not only one type, that is, copy paste is one type of plagiarism. There are different type of plagiarism, which I want. Sorry, time is not there. So these are the different aspects of research or scientific misconduct. Here I would like to say a few things about good scientific practice which we should try. Integrity, meticulousness, and accuracy is very important. Research, that is, it involves in all performing research, evaluating research, or recording, right? So maintain integrity, meticulousness, and accuracy. Ethically sustainable data collection is important. That means don't manipulate. Perform your research ethically. Evaluation method also should be ethical, and there should be openness in publishing. Taking due account of other researchers. That means you should not neglect. That is mainly concerned with your peer researchers. You should not undermine their value. Planning, conducting, and reporting according to standards set for scientific knowledge. Status, rights, authorship, liabilities, and obligations of the research team are determined and recorded in acceptable way. Very, very important, because it has been observed that, number of scientists, maybe from education institutions also, they adopt ghost authorship or guest authorship. That means they either remove the person who is involved in research from the authorship, or they intentionally add somebody else's name who has not been involved in the research. So that type of thing, that should not be done. Sources of funding and all that, although not very important for us. Publishing your research again is very, very important, but not touch this. So in nutshell, I just wanted to convey one or two very important messages, that drug research is nobody's prerogative, it is everybody's prerogative. Whosoever can contribute positively and constructively for the development of any drug. And for a good medicinal chemist, not only that you have to have a particular training in certain fields, you also have to be, correct in your conduct, that is morally and scientifically. You have to be sure that you are honest. Otherwise, let me tell you at the end one example. one researcher in one university, he or she, I should not tell that, published number of papers duplicating same work. At that time, the plagiarism software not available. And later on, it was found out that the researcher has published same thing in many journals. Not only the researcher was blacklisted, the university was blacklisted for a certain period of time for publishing in that particular group of journals. So that is a very big implication. You should remember that if you are caught in some scientific misconduct once, you may be blacklisted for whole of your life. So you must never trade into this particular blacklisted field. So thank you very much, and I wish that you will adopt some of my piece of advice, and this will be helpful to you. Thank you very much.
Thank you, sir, for your insightful talk. Sir has already summarized his talk, but still, for the benefit of the audience, I would like to say that sir stressed upon the multidisciplinary nature of the drug discovery effort, and that is why it is everyone's prerogative. Whoever is interested and serious about drug discovery research is free to take it up and progress it further for the benefit of the mankind. He also stressed on the various qualities that we teachers, the medicinal chemists, can inculcate in our students and display them ourselves so that our students are interested and become better medicinal chemists. Thank you for your insightful examples, sir, wherein from how you said from teaching itself, we get good ideas, and these ideas can be converted into research with a purposeful outcome. Sir also mentioned about the advancements in the various fields of spectroscopy, etc. And definitely it would be another session altogether to learn from sir about the advancements in spectroscopies and the NMR interpretation, etc. from sir. So we look forward to having another session with you, sir. At the end, I would like to say that yes, sir has rightly touched upon the ethics aspects, and many a times it is found that we at Indian universities, we are duplicating the work which is already published and which has been carried out in Western universities or institutes. So we need to develop our own ideas. We need to come up with our own ideas and go ahead with the publication. Somewhere where we are lacking is we develop the ideas, we get ideas, we work on them, but then we fall back while publishing, maybe due to some kind of laziness, or I don't know how to define it. Sir, we have some questions from the participants. I hope you don't mind me posing them to you.
Yes, please. Yes, please.
Yes, sir. Mr. Tirth Patel, he wants to know what are the ideal qualities of a successful medicinal chemist.
In fact, I have already defined this thing that you should have the basic traits in the form of having good knowledge about organic chemistry, good knowledge about spectroscopy, and if not good, at least working knowledge about the drug designing approaches, right? And along with that, I would say, see, you must be deadly honest. See, here, let me tell you one very important thing happened with me also. See, as far as the research in medicinal chemistry is concerned, two types of data you get. One type of data is from the structure of the compound, which you get in the form of spectra, right? So if you want to give a particular structure to a compound, nobody can deceive you because the spectra will themselves speak whether the structure is correct or not. But the problem is with the biological data. It is very difficult, to find out the originality of the biological data, whether it has not been fabricated or really fabricated. Now, here you have to depend on the input of your students. And let me tell you, I face one problem with one of the students, that the biological data which was given to me was later on found to be fabricated. But once I came to know that the data is fabricated, immediately I wrote to the journal that I want to withdraw this paper because the data is fabricated, and my research student has now divulged that the data is not correct. So here lies, the importance of your honesty. As a researcher, you have to be deadly honest. Let me tell you, see, finding duplicity in biological data is very, very difficult. But once it is found out, then you are gone, right? So medicinal chemist, basically being a hybrid of chemistry and biology, has to be double sure about the authenticity of the biological data. And to perform, because as far as your synthetic work is concerned, I don't think you will be able to perform good medicinal chemistry unless you have the basic things in synthetic chemistry, spectroscopy. To some extent, yes, you can compromise with the designing part, but of course, designing is important if you want to makes good. That means the desired type of compound in a short time period. Otherwise, basic qualities are that minimum knowledge of these three things and your honesty. Okay.
Okay, sir. The other question which is asked is, what is lacking in Indian institutes as compared to Western institutes that we are lacking behind or lagging behind in research?
See, I think everything is lacking here. You don't have good infrastructure. You don't have good input in the form of students, the poor quality of the students. And also, I would say the researchers themselves, right? And apart from them, even if, let us say there are, I don't say that good researchers are not there. Of course, they are they are short in number. But even if you have good researchers, good facilities, the problem is with the system. Our system is not conducive for performing or discovering drugs. Let me give another example here. I was telling about the discovery of chondrocuronium. When the chondrocuronium, at that time it was known as chandonium. Chandonium was published and patent was taken before publication, right? So one company approached Professor Harkishan Singh for taking the rights of chandonium so that they could launch a drug. I have not shown the structure of chandonium, because it is a sterile molecule. There are a number of stereoisomers, right? So one particular stereoisomer could easily have been synthesized, right? Chandonium was one isomer, optical isomer, and another was not reported by us. Now, this company, once they came to know that, it is difficult in Indian system to take the rights, what they did, immediately they made another isomer and put it into the market. See, system was like that. Within 1 year or 2 years of approaching Professor Harkishan Singh, there was a new neuromuscular blocking agent in the Western market, just a stereoisomer of chandonium. So somewhere our system is also lacking. Our system is also not conducive for drug research. Even if you make a drug, see, you'll be surprised that very, very, very few compounds are there which are of Indian origin. Let me tell you one compound which comes in the form of Saheli, which basically is a anti-estrogen compound or SCRM compound. This could come into the market because it was a discovery by Dr. Nityanand. And Dr. Nityanand, being the director of CDRI, was a very, very powerful person. He could get the work done through, his contacts. That's why it came into market. Otherwise, bringing a compound, an experimental compound into market as a drug, our system I think is very lethargic, is not very conducive. So system need to be changed. And that's why I said it is not a very, very costly affair as it is being projected. It should not also take that much time, but the system has to be conducive. They have to support the researcher so that, whatever finding they have, they can be put for clinical trials and can be put into the market as a drug. Right? A lot of things are left.
Thank you, sir. The last question here comes is, how can we improve ourselves to be more productive in research?
See, improving yourself is in your hand. Nobody can tell you that, you do this or you do that. You make a self-appraisal wherever you are lacking, you improve it. So it is a constant self-appraisal which will improve you, right? If you have the basic traits, basic knowledge, then you have to improve, you have to evaluate yourself and you will get the results. Self-discipline has to be there, self-appraisal has to be there. Okay? Hello? Some net problem with Dr. Anuradha.
Yes, sir. Just a minute.
Interest in the subject for among the students. What would you like to say on that, sir?
I'm sorry, I couldn't get you because your net was not stable.
Okay, so I repeat, like these days we find students, they are more interested in gaining a degree rather than gaining knowledge.
Yeah, that's correct. That is my observation also. See, as a teacher, I find that the current generation students, they are very, very intelligent. They are much more intelligent than we and you. But again, I will tell, they lack is dedication. They don't have that much dedication. If they can combine dedication with the level of intelligence they have, I would say that they will do wonders, because I realize that the type of intelligence they have, we don't possess. But I'm sorry to say that the amount of dedication we had is totally missing in this generation. Let me tell you that when I used to work in my PhD, minimum 12 hours standing on the bench was a routine feature. And sometimes even we used to sleep in the lab, because if we are running some column for purification, we have to sleep in the lab. And even sometimes we used to miss our dinner. So that type of dedication is lacking. If that dedication can be combined with the intelligence they have, I think they will do wonders like anything. That's a problem with the current generation. If they can improve their dedication and hard work, they will do wonders.
So thank you very much, sir, for your insightful talk. We have many young faculty members as well as students in our audience. So I hope they will be taking home some message from your talk and trying to have a self-appraisal at regular intervals and improve themselves. With this note, I would like to thank Yadav sir for his efforts and in delivering this talk and being with us today. I request Tejas sir to present a certificate of appreciation. Sir, kindly accept this certificate of appreciation on behalf of LM College of Pharmacy, the Research Committee and IQAC at LM College of Pharmacy. We are indeed very thankful to you, sir.
Yeah, thank you.
Thank you very much, sir. Thank you very much, Divya.
Yeah, thank you very much, ma'am, for nicely chairing the session. Now I would like to invite Dr. Tejas Dhamelia, sir, Assistant Professor, Department of Pharmaceutical Chemistry, to deliver a vote of thanks. Over to you, sir.
Yes, thank you, madam, for giving this opportunity. A warm and graceful afternoon to Honorable Professor M.R. Yadav, sir, Professor M.T. Chabaria, faculty members, participants, and my dear friends. It's a privilege to propose a vote of thanks and to acknowledge the contribution of those who work really hard to make this as a successful webinar which we used to celebrate, I shall say, the science of science in the spotlight. And this time we had an opportunity to hear Honorable Professor M.R. Yadav, sir. So on behalf of Research Committee LMCP and IQAC, first of all, I shall extend my sincere thanks to Professor M.R. Yadav, sir, that who spared his valuable time from his busiest schedule. Sir, we today had an opportunity to hear your thoughts on drug discovery, prerogative, beyond the examples you have covered, and the teachings, the learnings we could learn from you. That is, surely all of us are going to benefit in the future, especially considering the medicinal chemistry fields. We also extend our really heart of vote of thanks to Professor M.T. Chabaria, Principal LM College of Pharmacy, who really acted as a catalyst to finalize this event with the utmost care and the support. An event like this cannot happen overnight. The wheels for the same started rolling weeks ago. So we have been fortunate enough to be back with the help of the team, Professor Anuradha Gajjar, Dr. Ketan Ranch, Ms. Divya Teli, and many other committee members of the research committee and IQS. So our sincere gratitude also goes to all the participants and the faculty members who attended and have been benefited with this webinar. Last but not the least, a big thank you to all of you who made this event successful. So thank you very much. Thank you so much.